Welcome to the first session on carbohydrate metabolism, part 1, which is focusing on glycolysis, and we'll give you just an overall viewpoint before you get into all the details. So these are the learning objectives that will be covered in this session in just a general format. So what is glycolysis exactly? So this is the first step of metabolism to convert glucose to pyruvate. Most cells use this to make energy in other metabolic processes, but many cells like red blood cells can only do this to convert glucose into ATP and form energy for the cells. All of these reactions do occur in the cytoplasm that you see here, and in order to start glycolysis, you must invest 2 ATP molecules, shown here in the beginning, that will start the whole process, and then ultimately create the NADH substrate further down, and ultimately to pyruvate in 2 different mechanisms here. This is then going to go further into the Krebs cycle and create more energy. So let's focus on a couple of things here on where there's regulation in glycolysis. So glucose comes down, as you see here, and breaks down into 2 different pathways. But the ones in the circles are the major substrate level phosphorylation that regulates glycolysis. So the conversion of 1,3-BPG, which is 1,3-biphosphoglycerate, to 3-phosphoglycerate, as shown here in the first circle, and the second circle, which is the conversion of phosphoenolpyruvate to pyruvate by pyruvate kinase. There are also 3 rate-limiting regulated steps. The initial first one, which is glucose to glucose 6-phosphate, the second one, which is fructose 6-phosphate to fructose 1,6-phosphate, and then the third one is the phosphoenolpyruvate to pyruvate at the very end. Hexokinase and glucokinase are the first 2 starting points. These enzymes are mainly found in the liver, which is why the liver does most of the glycolysis for the whole body. These are irreversible, and it traps the glucose into the cytoplasm, which then goes further downstream. So hexokinase and glucokinase are inhibited by their end product, which is glucose 6-phosphate, and it is induced by insulin to start this process. Glucokinase metabolizes sugar at very high concentrations. It's not really inhibited by the glucose 6-phosphate, so it can continue to break down glucose. And again, insulin is a big regulator of this part here. As we move down, glucokinase is mainly in the pancreas, and it is supposed to be influenced by, it senses the glucose is sensed by the glucokinase, and that's what turns it on and then brings the glucose in and starts the glycolysis process. The advantage of the liver is that dietary glucose can go straight to peripheral tissues instead of being trapped as the glucose 6-phosphate. So if you have a very low glucose, this will inhibit this process, and the glucose will stay in the plasma so it can be used by tissues other than the liver. But if you have a very high glucose, then that shifts more of this into the liver to further go down into glycolysis. So the next part of this is phosphofructokinase, which is converting fructose 6-phosphate to fructose 1,6-bisphosphate. Now the reverse process of glycolysis is gluconeogenesis, which we'll discuss in a different video. So this process here, once PFK does its thing, is irreversible. And it basically says glucose now has to go through the rest of glycolysis and become pyruvate. This PFK1 is also activated by fructose 2,6-bisphosphate, which is another byproduct here of fructose 6-phosphate. So it converts and just keeps going in this cycle to activate it like this, and further propagates glycolysis. The FBPase2 is what catalyzes the reaction of this fructose 2,6-bisphosphate, the enzyme fructose bisphosphatase, to the fructose 6-phosphate. So this will be a regulating step here. If you're making too much of this product, it will go back and start inhibiting further production, so that this is one regulatory step that is there in glycolysis. And this is partly regulated by PFK2 versus PFK1, which is taking that product down into glycolysis further down. So PFK2 kind of serves as a regulatory step and a buffer to control how much is going down when necessary. Glucagon is going to hugely regulate glycolysis. in the sense that glucagon activates it. And it's telling the enzymes cyclic CMP, which activates some of these other phosphorylations, and that deactivates the FBBase2 so that it lets this go further down. Insulin, on the other hand, is going to result in higher activity of PFK2 and increased fructose 2,6-bisphosphate concentrations, which will then regulate that step a little bit there. So epinephrine is the major predominant hormonal influence, just like glucagon. It also can increase cyclic AMP, which leads to increased activity of the phosphofructokinase enzymes. This pKa then phosphorylates different parts of these enzymes, so it leads to increased fructose 2,6-bisphosphate and an increased rate of glycolysis. The final step that we see in glycolysis as it goes down is pyruvate kinase. That's the next major step that you should learn. So these 3 points are the major learning points for glycolysis. Hexokinase, glucokinase, as we mentioned, that actually brings glucose in. And then phosphofructokinase, which is in controlling this fructose part. Once it gets down into these steps in a DHAP or glyceraldehyde, these next few steps go fairly quickly, but they're also reversible for gluconeogenesis. But this final step of pyruvate kinase is what makes pyruvate, which is the end product of glycolysis. Glucagon, again, causes more phosphorylation, so it prevents this to happen faster because it's going to want glucose released into the blood, so it'll activate the reverse process. Insulin is more anabolic, so it's going to stimulate this process further down and form more ATP. This concludes this session. If you have any questions, please subscribe to the YouTube channel to get new updates on further new content. Thank you.